Imagine you just finished a really grueling 20-minute workout.
Like a heavy cycling session or a quick HIIT circuit?
Exactly. You are completely sweating, your heart is just pounding against your ribs, and, your muscles are incredibly heavy with fatigue.
Yeah, that classic post-workout burn.
Right. And you are probably just thinking about the calories you just burned or waiting for that endorphin rush to kick in. But deep inside your quadriceps, beneath the muscle fibers and down into the microscopic nucleus of your cells, something much more profound just happened.
Something most people do not even realize is going on.
Exactly.
Ugh.
Because in those 20 minutes, you literally physically broke the molecular locks on your own DNA.
Which is just wild. I mean, that image completely upends the way most of us were taught biology, right?
100%.
We are usually handed this idea that your genetic code is essentially like a stone tablet, just a static, unchangeable blueprint that you inherit the day you were born.
Right. The whole narrative that you were dealt a genetic hand and, well, your only option is to just play those cards the best you can.
Exactly. But the science is showing that reality is far more dynamic than that.
Today, we are exploring a really counterintuitive paradox. Your actual genetic code, the actual sequence of letters, might be the exact same as it was the day you were born, but the way your body reads that code can be radically rewritten in a matter of minutes.
And it does not take years of dieting or crazy interventions to do it.
No, it doesn't. Our mission for today's deep dive is to bridge the gap between the physical act of just moving your body and the deep, complex molecular biology happening inside your cells. We are looking at a landmark study published in the journal Cell Metabolism.
Oh, this is a phenomenal paper by researchers from the Karolinska Institute and the University of Copenhagen.
It really is. They discovered exactly how a single, standard bout of exercise physically remodels your epigenome. But to really grasp the sheer scale of this discovery, we have to look at how epigenetics actually control the body in the first place.
Right, because every single cell in your body contains the exact same massive, comprehensive library of DNA.
Yeah, your eye cells, your liver cells, your muscle cells, they all hold the exact same instruction manuals.
Which is a problem if you think about it.
Exactly, because a muscle cell absolutely does not need to read the manual on how to, produce stomach acid or grow hair.
If every cell tried to read every book at the exact same time, it would be biological chaos.
Total chaos. So the body uses a process called DNA methylation to keep order. Let's try to visualize this. Think of your DNA as a giant library, and think of these methyl groups not just as padlocks on a door, but as thick molecular superglue binding the pages of specific books together.
That is a perfect way to visualize it, because the cell has transcription machinery. You can think of it as a biological photocopier that travels along the DNA strand, trying to read the recipe to build a protein.
Okay, so it's scanning the pages.
Right, but when a gene is heavily methylated, those pages are glued shut. The photocopier physically cannot pry the pages apart to read the instructions, so that specific gene remains completely silenced.
And historically, we have always been told that this superglue is incredibly stubborn, haven't we?
Oh yeah. The broad scientific consensus for a long time was that DNA methylation was mostly mitotically stable.
Meaning it doesn't just change overnight.
Exactly. The assumption was that, sure, decades of smoking or, a lifetime of specific dietary habits might slowly add or remove some of this glue, but it was viewed as a very slow, generational, or long-term environmental process.
Wait, so we are taught that epigenetics are long-term, environmentally driven changes. Are you telling me that we aren't permanently stuck with the genetic padlocks we were born with, and that a single normal workout can literally pick those locks?
That is exactly what the researchers at Karolinska and Copenhagen set out to prove. They challenged that old assumption directly. They hypothesized that the superglue wasn't permanent at all in skeletal muscle. Yeah, they believed that a single everyday workout could act as a sort of chemical solvent, rapidly dissolving that glue to activate specific metabolic genes on demand.
But subjective feelings aren't data, right? You cannot just ask someone if their DNA feels different after a session on a stationary bike.
Definitely not.
To prove this, you would literally have to look at the muscle fibers before the sweat even dries, without, completely disrupting the very tissue you were trying to study. How did they actually pull that off?
Well, they designed an incredibly rigorous human experiment. They recruited a cohort of healthy, young, sedentary men and women.
And the key word there is sedentary, right?
Absolutely. They specifically avoided elite athletes because a highly trained athlete's muscles are already vastly adapted to constant physical stress.
They needed a clean slate.
Hmm.
People whose metabolic genes were still, like, firmly glued shut from a general lack of intense physical demand.
Exactly the right starting point. So they brought these volunteers into the lab and took a baseline muscle biopsy from their quadriceps, specifically targeting the vastus latalis muscle on the outside of the thigh.
Ouch.
Yeah, they used a specialized needle to extract a tiny core of actual muscle tissue. Then they had the subjects undergo a single acute bout of exercise on a cycle ergometer.
Just a stationary bike.
Right, and about 20 minutes after the workout finished, they went back in and took a second biopsy from that exact same muscle group.
I really have to pause here and just appreciate the immense commitment of those volunteers. I mean, donating a chunk of your thigh muscle, doing a grueling cycling test, and then immediately letting researchers take another chunk of your thigh is just a massive contribution to science.
It absolutely is. And their effort yielded incredible data. The researchers now had a literal before and after snapshot of the muscle tissue at the molecular level.
That is amazing.
It is. And to analyze those tissue samples, they deployed a couple of highly sensitive molecular techniques. The first was the LUMA assay.
LUMA. Okay, how does that
Okay.
work?
You can think of LUMA as a global molecular barcode scanner. It sweeps across the entire genome and counts the total amount of methyl superglue present across the entire DNA strand. It gives a macro-level snapshot of methylation.
And what did that global scanner reveal when they actually compared the pre-workout muscle to the post-workout muscle?
The global DNA methylation significantly decreased.
Just like that.
Just like that. The superglue was dissolving across the board. But, a global sweep only tells you that a broad change happened. It doesn't tell you what the change actually means for the human body.
Right, it's just a raw number.
Exactly. So they used a second, much more targeted technique called MEDP-qPCR.
Okay, so if LUMA is the global scanner, MEDP qPCR is like the magnifying glass. It lets them zoom in to see exactly which specific books in the biological library were being unglued.
That targeted search is where the paper becomes truly groundbreaking. They found that the methyl glue was specifically being stripped away from the promoter regions of vital metabolic genes.
Specific genes. Which ones?
We are talking about genes with highly technical names like PGC1 alpha, PDK4, and PPAR delta.
Okay, let's demystify that alphabet soup, because understanding what those specific genes actually do is the key to why this study matters to anyone listening. Why should we care that PGC1 alpha is suddenly accessible to the cell's photocopier?
Because PGC1 alpha is essentially the absolute holy grail of mitochondrial biogenesis.
The Holy Grail.
Yes, it is the master regulator gene that tells your cells to build more mitochondria. And as a reminder, mitochondria are the microscopic power plants inside your cells that convert the food you eat and the oxygen you breathe into usable ATP energy.
So when you unglue PGC1 alpha, you're basically telling the muscle to build a bigger, more efficient engine.
Exactly. And meanwhile, PDK4 and PPA delta govern how your muscle actually uses fuel. They specifically shift the balance to optimize burning stored fat for sustained energy.
Wow. So the physical act of pedaling the bike dissolved the glue specifically on the instruction manuals for building better energy factories and burning fat more efficiently.
Yes, very specific, targeted adaptations.
And the researchers saw the immediate downstream effect of this, right? Because ungluing the book doesn't matter if nobody actually reads it.
You are spot on. The downstream data perfectly validated the mechanism. As the methylation levels dropped as the pages were pried apart, the researchers measured a massive spike in mRNA expression for these specific genes.
And mRNA is the messenger.
Right. It means the cellular photocopier had successfully attached to the newly unglued DNA, read the instructions, and was actively churning out the blueprints to build those new metabolic engines. And the entire sequence happened in a matter of minutes.
This speed really challenges everything we were taught about genetic adaptation. But delving deeper into the methodology, this is not just a binary on-off switch where, like, any random movement triggers the ungluing, is it?
Not at all.
The researchers ran a second cohort experiment to look at the actual intensity of the exercise. And the dose-dependent response they found is arguably the most crucial part of the whole study.
I agree, because it shifts the conversation from pure biology to practical application. So they took a new group of subjects and had them exercise at two distinct intensities.
Okay.
First was 40% of their maximal aerobic capacity, which is a light conversational pace where you might break a mild sweat but can still breathe totally comfortably.
Like a brisk walk or a light jog.
Exactly. And the second was 80% of their maximal capacity. At 80%, you are really pushing your limits, your lungs are burning, and you definitely cannot hold a casual conversation.
And the cellular response was drastically different between the two.
Completely different. When the subjects pushed to that high-intensity 80% threshold, the global scanner and the targeted magnifying glass showed an immediate, profound drop in the methylation for those metabolic genes the moment the workout ended.
The glue melted right away.
Yes, but at the 40% low-intensity pace, the ungluing effect was incredibly muted. It was either significantly less pronounced or it was delayed by several hours.
Why is that? What's going on physiologically?
Well, the underlying physiology explains it perfectly. When you are lightly jogging at 40% capacity, your body only recruits a fraction of its available muscle fibers, primarily the slow twitch fibers that are highly efficient but very low in power output.
Makes sense.
But when you demand 80% output, your central nervous system essentially panics. It fires everything it has, recruiting massive bundles of fast-twitch muscle fibers and demanding an immense, localized metabolic response.
Which raises the ultimate mechanistic question, right? How does the muscle cell actually know it is working at 80%? What is the actual biological messenger that travels from the physical flexing of the quadricep all the way down through the cell membrane into the nucleus to tell the DNA to dissolve the superglue?
That is the million-dollar question. And to isolate that exact messenger, the researchers had to strip away the complexity of the human body.
What do you mean?
They couldn't use human subjects on a bike anymore because a living human is, well, messy. When you exercise at 80%, your brain releases adrenaline, your cortisol spikes, your overall blood flow changes.
There's too much biological noise.
Exactly. They needed to prove that the DNA change was caused purely by the mechanical contraction of the muscle itself, independent of all those systemic human hormones.
So they took the experiment out of the human and into the petri dish.
They did. They utilized isolated mouse soleus muscles and cultured rat myotubes, specifically a cell line called L6.
Okay, rat muscle cells.
Right. By keeping these muscle cells alive in a highly controlled laboratory environment, they completely eliminated the nervous system and hormonal noise. It was just the muscle tissue in isolation.
But then they faced a completely new problem. How do you simulate an 80% VO2 max sprint in a tiny dish of isolated cells?
And their solution is honestly one of the most fascinating chemical tricks in the entire paper. They expose these rat muscle cells to heavy doses of caffeine.
Wait, caffeine?
Yes, caffeine. In a highly concentrated in vitro laboratory setting, caffeine acts on a very specific cellular structure called the sarcoplasmic reticulum.
The sarcoplasmic reticulum. That sounds intense. What is that?
You can think of it as a tightly sealed internal storage vault that sits inside every single muscle fiber. And inside that vault is a massive concentration of calcium ions.
Okay, I'll say.
In human physiology, when a nerve impulse hits your muscle and tells it to contract, it essentially blows the doors off that vault, allowing an absolute tsunami of calcium to flood the main body of the cell. That calcium physically binds to the muscle proteins and causes the physical contraction.
So a harder, heavier contraction at 80% intensity means a vastly larger and more sustained tsunami of calcium flooding the cell compared to just a light jog.
Exactly.
So the researchers hypothesized that calcium wasn't just causing the physical flex, it was actually the messenger traveling to the nucleus.
And the caffeine application proved it beautifully. The high dose of caffeine forced the sarcoplasmic reticulum in the rat cells to dump its calcium into the cell, perfectly mimicking the massive calcium flux of a high-intensity workout.
And what happened to the DNA?
The result was rapid, profound hypomethylation. The superglue dissolved, the metabolic genes were fully exposed.
Okay, hold on. Let me just push back on this for a second because my brain immediately goes to a very lazy, life-hacking place.
I think I know where you're going with this.
If caffeine forces the calcium vault to open and unglues the DNA, does that mean me sitting on my couch sipping a massive double-shot morning espresso is secretly giving my muscles a genetic workout? Can I just caffeinate my way to mitochondrial biogenesis?
It is a brilliant question, but sadly, human physiology completely thwarts the lazy approach here.
Yeah.
Yeah, you absolutely cannot caffeinate your way to genetic fitness. In a living, breathing human body, drinking coffee does not create the massive, localized calcium flux inside your skeletal muscle tissue that a physical contraction does.
Why not?
Your stomach absorbs the coffee, the caffeine hits your central nervous system, and you feel mentally alert. But the dose is nowhere near high enough to pry open the calcium vaults in your quadriceps. The in vitro caffeine experiment was strictly a chemical skeleton key used by the researchers to prove the signaling pathway in a dish.
Ah, okay. So the true catalyst still absolutely requires the mechanical work. You need the physical stress and the localized calcium flooding that only comes from physically moving the muscle under an actual load.
Yes. And the researchers cemented this fact with one final test. They took those same rat cells and applied a drug called dantrolene.
What does dantrolene do?
Dantrolene's primary function is to lock the doors of the calcium vault, preventing any calcium from escaping, even when the cell is strongly stimulated.
Oh, wow.
And when they applied the calcium blocker, the entire epigenetic process ground to a dead halt. The DNA remained firmly glued shut, and the gene expression stopped completely. No calcium tsunami means no genetic remodeling.
That is incredibly clear. The mechanism is undeniably proven. The physical contraction triggers the calcium flood, the calcium acts as the messenger to the nucleus, the methyl glue dissolves, and the metabolic genes are activated. The causality is just beautiful.
It is elegant biology.
But taking this dense cell metabolism paper out of the lab, how do we translate this into optimizing our daily routines? If calcium flux and exercise intensity control our genetic expression, what are the concrete actions someone listening today can actually take?
Well, the science points to a few very clear, actionable protocols that leverage these exact cellular mechanisms. Protocol 1 is prioritizing high-intensity aerobic thresholds over sheer duration.
This directly addresses the biggest hurdle most people face, which is just a lack of time.
Exactly.
We so often convince ourselves that if we do not have 45 minutes for a proper, long jog, there's just no point in exercising at all. But the 80% versus 40% dose-dependent response shatters that excuse entirely.
Completely shattered.
If you only have 15 minutes, pushing your body to that 80% capacity where your breathing is labored and holding a conversation is basically impossible, is the scientifically proven way to immediately force the epigenetic locks open on your mitochondrial genes.
You are manipulating the cellular mechanics in your favor. It is the difference between waiting hours for a low-intensity 40% workout to maybe trigger a delayed, mild genetic response versus generating a massive calcium tsunami that strips the methyl groups on demand.
Right.
If time is your enemy, intensity is your biological skeleton key.
I love that phrasing.
Yeah.
But biological adaptations come with a hidden cost, which brings us to Protocol 2. Let's call it the transient consistency rule.
Yes, this is a tough pill to swallow.
Buried in the data of this study was an observation that initially sounds incredibly demoralizing. The researchers noted that 48 hours after a rigorous 3-week training program ended, the DNA methylation levels in the subjects returned completely to baseline.
The superglue came right back. The metabolic genes were silenced once again.
Wait, so all that excruciating hard work at 80% VO2 max is completely erased in just 2 days? Why would human biology sabotage us like that? It honestly feels like a design flaw to board up the windows the second you stop working out.
It feels like sabotage in the modern world, for sure. But from an evolutionary biology perspective, it is a brilliant survival mechanism.
How so?
Maintaining an abundance of highly active mitochondria and hyper-efficient fat-burning pathways requires a massive amount of baseline cellular energy. Thousands of years ago, if you weren't actively fleeing predators or hunting for food, if the physical stressor just disappeared, your body assumed you were entering a period of rest or potential famine.
Oh, I see.
So it aggressively boarded up those metabolic windows and brought the superglue back to conserve calories.
The body just refuses to keep expensive biological machinery running if the environment doesn't explicitly demand it.
Exactly.
The actionable lesson here is really profound. Epigenetic unlocking is entirely transient. You are not building a permanent house. You are essentially just paying rent on your genetic fitness.
That is exactly it. You cannot exercise intensely for a month, get into phenomenal shape, and assume you have permanently reprogrammed your DNA. The biological clock is always ticking toward baseline. The hack is recognizing the reality of this 48-hour window. You have to consistently re-expose your cells to that intense mechanical stress every day or two to keep the calcium flowing and the metabolic pathways unglued.
It completely reframes why consistency matters so much over sporadic hero efforts in the gym.
It does.
Which leads perfectly into Protocol 3, the contraction trigger. because telling someone they need to repeatedly push their physical limits every 48 hours can sound pretty exhausting.
It sounds like a lot of work.
But we need to look back at how the researchers handled the isolated mouse muscle. To trigger the hypomethylation without destroying the tiny isolated muscle, they used simulated contractions consisting of just 0.3-second electrical impulses.
Right, they were not simulating a marathon in the petri dish. They were simulating brief, intense bursts of mechanical tension.
And the hack here is realizing you don't necessarily need to change into workout gear and go to the gym to communicate with your DNA.
Got it all.
Brief, intense bouts of muscle contraction are sufficient to trigger that localized calcium release. If you are stuck at work or working from home, a single minute of rapid, deep air squats or, an all-out sprint up 3 flights of stairs acts as a contraction trigger.
You are intentionally generating the calcium flux in the largest muscle groups of your body. It lowers the barrier to entry significantly. You can slip these epigenetic triggers into the margins of your workday.
Just sneaking them in.
Every time you contract a muscle intensely, you are actively participating in a chemical conversation with your own genome.
That is just incredible. As we wrap up this exploration of the cell metabolism paper, I want to leave you with a final provocative thought. You're not a passive victim of a static genetic blueprint. You are the active daily architect of your own gene expression.
That is the big takeaway.
The hardware you were born with is fixed, sure, but you are completely in control of the software. Every single time you push your physical intensity, you are generating the chemical messengers that physically alter the shape of your DNA, building a better, more resilient metabolic engine. Your daily physical habits are literally the pen that writes your biological reality.
It entirely shifts the mindset of exercise from simply burning calories to literally managing the molecular health of your cells.
It really does. Now, we have spent this entire deep dive exploring how intense physical output alters the DNA inside your muscles to boost your energy and efficiency. But what happens when the workout ends? What about the fuel and the chemical building blocks you consume to recover?
That is where things get really controversial.
Right. If you spend any time around fitness culture, you probably think that drinking branched-chain amino acids, BCAAs, after a heavy session is simply providing the raw materials to repair the biceps or quads you just broke down.
That is the standard, unquestioned narrative across the entire supplement industry. You break the muscle, you feed it amino acids, it grows back stronger.
But there is a bizarre, deeply counterintuitive paradox hiding in your post-workout shake. What if those exact same amino acids, aggressively marketed for your muscles, are actually traveling up your bloodstream and hijacking the highly sensitive transport gates in your brain?
Oh, this is a fascinating topic.
What if your physical recovery protocol is secretly altering your mood, spiking your mental fatigue, and shifting the delicate chemical balance inside your head?
When you look at the pharmacology of how amino acids cross the blood-brain barrier, it really makes you question everything you thought you knew about standard sports nutrition.
Next time, we are diving into exactly that, how BCAAs impact your brain, the science of amino acid transport. Get ready to ponder how the supplements fueling your fitness routine might be secretly running your brain chemistry. Until then, keep picking those genetic locks.